Now, for better understanding of the membrane transport, we are dealing with facilitated diffusion over here. Let us understand a few more properties of the proteins, carrier proteins, which are present through and through across the fluid membrane, that is the lipid bilayer. We have to understand that these proteins play a crucial role. They are very important for the transport of the particles across the membrane, and these are known as porins. As the name suggests, porins is derived from pores. They are having pores present in them. Now, what are these porins? They are nothing but carrier proteins, alright. Now, these carrier proteins, they will have carrier or channel proteins. They will have a channel present inside them through which the particular particle would move across the concentration gradient. Supposedly, this is the carrier protein I'm talking about. It will have a pore inside it, which is having a channel inside it. This would be the porin. Now, porins are present extensively in the membrane of mitochondria, because many enzymes are required for movement across the mitochondria. We have discussed that. Now, these porins, they can enable the movement of proteins. Such big are the pores that they can enable the movement of the proteins, large proteins through them. And the movement that we study, that is taking place, that is further classified into symport, antiport, and uniport. Okay, this is the movement across the carrier proteins through the porins. I must tell you, porins are the simple proteins, and one thing more you have to remember, that there are large proteins for movement of water molecules across the membrane, which are known as aquaporins. This is just to be crammed directly, that aquaporins are those carrier proteins which would enable the movement of the water molecules across the fluid mosaic membrane that we study, because it is lipid. Lipid is mostly, as you know, they are not much friendly, they are not philic towards water, so that is why there are aquaporins which are present. And apart from that, not all the biomolecules would be lipid friendly, as I must say. Whichsoever would be hydrophilic, that would also be resisted by the lipid. That is why we have these carrier proteins, which are also known as porins, and the movement across them, let us study one by one. Let us first of all take uniport into account. Now, what is in the case of uniport? Supposedly this particle, understand it from here. Supposedly this particle I'm talking about, this one, okay, this is going across one direction only. That means at a time only one particle would be moving across the pore of the carrier protein. That would be known as uniport. Uni means single and movement is uniport. That means only a single, it could be protein molecule or any other particle which has to move across the proteins, carrier protein, that would be termed as uniport. Now, at times what happens, suppose there are two particles, A and B particle I'm talking about, and they are moving simultaneously inside, two at a time, that would be known as symport. Okay, uniport was a single, as in that case you can see. Symport would be two molecules, two different molecules would be moving through the carrier protein at the same time. That would be termed as symport. And consider that this B particle is present inside the cell and it has to move outside, and the A particle has to come inside. That would be antiport, because both the particles, they are moving in opposite directions, as you must have seen if some of you have ever traveled in train. When two trains are crossing each other, same thing happens over here. Okay, at times two trains are running parallel, or you can take a car's example. One car is coming from front direction, you're moving like this, so they will cross at one point, that would be antiport. If they are moving in similar direction, that would be symport. And supposedly your vehicle is running all alone on the entire road, that would be uniport. So this is how you understand the movement of the particles across the carrier proteins in terms of the terms symport, antiport and uniport through the porins. Alright, thank you.